And they're also now doing work on just harvested lymphocytes from non-related donors and just using them and trying to do the same thing. But that's not where the hope is. The hope is we'll be able to get it from the actual patients themselves.
Do they talk about any difference in mechanism or problems with solid tumor cells?
Well, they haven't done a lot of research in solid tumor cells because they don't have markers that are as good. So this technology is really as good as the marker. So since CD19 and CD22 pretty much get all B cells between the two of them, it's really good for B-cell lymphomas and B-cell leukemias. But the other tumors that have good markers, the theory is that the technology is going to work for them too. But it's really dependent on the markers on the lymph or on the cancer cells, how specific they are for that cancer, how specific they can make these these CAR T cells to go after them. And again, David probably knows more than I do.
I know that they're trying to target CEA. There are different things that they try to do with that. So another T cell therapy that they are working on is anti-HIV, and they insert this CAR construct into the CCR5 locus, so they will be protected from HIV themselves, and then trying to target other antigen on HIV-infected T cells. So HIV is another one. For solid tumors, I know CA is one of them. And there are recent studies trying to engineer Tregs and induce tolerance. So there are two animal models, one with an IBD model and another one of EAE model, MS-like model. And it seems to be effective as well in designing the Tregs to recognize specific antigens related to the disease and just controlling the disease with CAR Tregs. So yeah, they're trying everything they can with those CARs.
It's a pretty significant change in our approach to cancer. That's what I was thinking.
It started actually in the late 80s.
Yeah, but it didn't really go big time until these last couple of years. Rupp's really pushing this from Philadelphia. He's sort of the beginning of all this. And he's pushing this really strongly now, is trying to get everybody involved to come up with other markers. And again, you can see the utility of this. If you can put any CD marker on this cell and just wipe out that particular cell lineage, I mean, it just as soon as we get good markers, we should have really good tumor therapy for pretty much all those diseases.
And with the cytokine storm, I think there was one paper in New England 2 years ago, and after one dose of anti-IL-6, everything went away.
Right.
Great responsive treatment.
So you can add your $15,000 drug to prevent your $20,000 side effect. But it works really well, and that's the thing that's nice. Anyway, I didn't do two. I thought I'd do the last one.
Oh, yeah. I'm just curious if there's any protocols looking at this, like right after conventional therapy, or is the primary therapy usually so good? In other words, like ALL in kids at 90+%.
We're getting 80 to 90% with primary therapy, and these are all pretty much studies in phase 1. They have to be basically failures of primary therapy to get into the studies. And the other ones, like neuroblastoma and pancreatic, since those people don't have any time, they're not putting the same requirements on them. All they have to do is have a diagnosis, and then they have to either be at Memorial Sloan Kettering or at the NCI, because that's the only place they're doing adult studies right now with those solid tumors.
Well, did you run into any cost estimates?
I just wondered about that myself, like what would this stuff cost? I mean, harvesting the T cells would cost a few $1,000s right there. And then what these guys do at Creative Biolabs, I don't know whether they all drive Teslas or what they all drive at Creative Biolabs, but I don't think they're losing money. So, but I know there was not even a mention of how much it costs. So, Dave, you got any idea there? I would imagine it's not cheap. That's what I would imagine. But anyway, it's just something you're going to hear about in the future, so now you've at least heard about it. So, I don't have anything else.
Right here, buzz it on.
Excellent.
Anyone else? It sounds like traffic's bad out there, so we should always head out.
Going north, it's not bad. It's only coming south.